Hi, and welcome to our GMO Lab webinar. I'm Dr. Katy Martin. I'm a member of the Mini PCR Bio team, and I'm really excited to be sharing this lab with you today. I'm joined by my colleagues in the chat section. If you have any questions today, feel free to drop them there. We'll do our best to get you an answer. Now, I was a classroom teacher before I joined the Mini PCR Bio team. And so I'm really excited to share this particular lab with you today because it's the exact type of lab that I most love to do with my students. And that's because it's a lab that is deeply connected to a real-world issue that students hear about all the time. In this case today, we're talking about GMOs, or genetically modified organisms. We hear about GMOs all the time on the news. We read about them in the headlines. They might, whether or not a food contains GMOs might influence our dietary decisions. This is a really relevant issue, but yet it's one where there's a surprising lack of understanding of the core technology at the centerpiece of this issue. So the quotes on this slide are quotes I pulled from a 1997 study of consumer attitudes about GMOs, but they may as well have come out of my own students' mouths. So I would hear students saying things like, oh, this food's been messed with in a laboratory. I find the concept of genetic engineering of food scary. And in general, there's a lack of understanding around what is being put in a GMO food to genetically change it. What is a GMO at its core? And the exciting thing about this lab is that it's going to answer exactly that question. So when we're done today, we'll know exactly what a GMO is. What is that technology that's at the core of a GMO? So what we're going to be doing today is testing grocery store food that I bought at the market down the street for the presence of transgenic elements indicative of genetic modification. This is going to help us understand how desired traits can be introduced in plants through genetic engineering. And if you do this lab in your classroom, it serves as a great platform for an evidence-based discussion about genetic modification, and your students are going to be collecting that evidence themselves by doing the lab today. So I think when we're talking about GMOs, it can help to back up a step and talk a little bit about the context in which they emerged. So long before GMOs, humans began selectively breeding plants for traits that promoted a better and more robust food supply. So we would select for things like the ability to grow in a wider variety of environmental conditions, or for big, juicy, nutritious fruits. The illustration I have on this slide is an example of the latter. Does anyone know what modern crop descended from teosinte? Believe it or not, corn. Modern corn is the modern day version of teosinte. By selecting for big grains and juicy starch-packed kernels, humans were able to kind of coax teosinte to eventually become modern day corn. And modern day corn is a really nutritionally dense food compared to ancient teosinte. Now, this process of selective breeding is a form of genetics. It's a genetic practice. So farmers were acting on observable phenotypes, but in selectively breeding for particular phenotypes, they were acting on the underlying genotypes as well. Now, in the last century, as scientists have developed a more sophisticated understanding of genetics, they started to explore ways they could directly change a plant's genome without first needing to, selectively breed for generation upon generation. Their early methods were crude, so they might blast a plant with radiation to increase its mutation rate and thus increase the rate at which desirable phenotypes emerged. Since then, though, over time, their methods became more and more precise, and GMOs are one example of that. So when we talk about GMOs, this is what we're talking about. We're talking about taking a gene that you find in one organism that confers a desirable trait and transferring it into another organism that doesn't natively bear that gene. So once we've done that, once we've taken this desirable gene and transplanted it into our other organism, we would say that organism 1 is now a GMO. It has been genetically modified. And we would call that gene that we borrowed a GMO transgene. And this GMO transgene is something we can test for using modern molecular biology tools that might be common in your laboratory. And so that's what we're going to be doing today. We're going to be testing for the presence of this GMO transgene using a common GMO marker that I'll explain a little bit more later. And we're going to be doing that through a 3-step experiment. We're going to begin by extracting DNA from our foods of choice. We're going to amplify that GMO transgene with PCR, and then we're going to use gel electrophoresis to visualize whether our food contained that gene or not. Now, if you're doing this in a classroom, this is a 2-day lab. You do DNA extraction and PCR the first day, and then the second day you'd come back and do gel electrophoresis. I am going to breeze through all this in just about 45 minutes, cooking show style today. So let's begin. We're going to extract some DNA from our foods of choice. The beauty of this lab is that you decide what foods you're testing. You can go to the grocery store and grab fresh foods like strawberries, bananas, and corn, but believe it or not, you can also extract DNA from processed foods like chips, like veggie burgers, like flour. And in fact, those foods are easier to extract DNA from than fresh foods. So this is a table that we publish with our lab. It's kind of a cheat sheet for you. It tells you what foods it's relatively easy to extract DNA from and which ones are best to avoid. And you'll see our top row here. The processed foods tend to yield the most robust results. Fresh foods, the kind of rule of thumb is that the wetter the part of the plant you use, the worse your results are going to be. So if you're doing a strawberry, say, you want to sample the stem or leaf of the strawberry and not the big juicy edible fruit itself. When you sample from like the wet part, there's usually so much water that there's not a ton of DNA to get out of that part of the plant. So today, for our purposes, I'm going to be looking at 2 types of corn chips. So I'm going to be testing our Brand X corn chip, very popular corn chip that on its packaging makes no claims about whether or not it is a GMO or contains GMO ingredients. And then I'm also going to be testing my Brand Y corn chip. Now, this corn chip bears this label that claims that it is non-GMO project verified, so it should not contain any GM ingredients. And we're going to put that claim to the test today in our lab. So let's begin. When we take a sample of a processed food, what we want to work with is a really small amount. You just want to take a crumb of your processed food, or a really tiny little portion. Now, we're talking like a 1 to 2 millimeters in diameter for a crumb. I'll show you just how small my portion is here. I have this blue corn Brand Y chip. I'm going to put it in an orange bowl so you can see it under my webcam here. Just a really tiny fragment. Now I'm going to take that crumb and I'm going to put it in 50 microliters of lysis buffer, or my DNA extraction buffer. And so I've already pipetted some extraction buffer into a tube before we started today. I'm just going to drop my crumb now into that tube. You can see it at the bottom of the tube there. Now, as I prepare my other corn chip to sample, I'll just make a note about extracting DNA from fresh foods, which we're not doing today, but if you wanted to do it, what you'd want to do is, again, take a leaf or a stem from that plant, and then you'd want to take a pipette tip and puncture that tissue a few times until a bit of tissue clings to your pipette tip, and then pipette up and down into your DNA extraction buffer. You just want a tiny, tiny sample. That's kind of counterintuitive. You might think the more tissue, the better, or the more of my chip, the better. There's more DNA, the more you sample, right? But too much can actually inhibit your PCR reaction later on, so you really just want a teeny, tiny bit. All right, so we have our food samples hanging out in our DNA extraction buffer. This DNA extraction buffer is an alkaline buffer, so it's really high pH. It's going to bust the cell walls open, release the DNA inside. It's going to do that under high heat. So we want to heat up our samples now. You can do this in a hot water bath or with a heat block. I'm actually going to be using my mini PCR to heat these samples up today. So this is the same machine I'll be using later on for PCR, but it can also be used as a heat block. So let's set our samples up in there now. So I'm going to switch to my webcam. Here's my mini PCR, and I'm just going to open the lid and put my 2 samples in, close the lid and make sure it's tight, and then we're ready to go. Now, the mini PCR you connect to and control with your own device using our downloadable app. So I'm going to click over now to my mini PCR app and program a new heat block. So we're going to call this 95-degree heat block, and 95 degrees for 5 minutes will do it. We'll save and run that, and it'll get started. Okay. Now, as our DNA is being extracted, let's talk about the next step of our protocol, which is PCR. So if you're not already familiar with PCR, PCR stands for polymerase chain reaction, and it's a way that we can replicate a specific DNA target. So we start with a complex DNA sample. So think about what we extracted from our chips. It's going to contain all the corn genes, non-coding sequences from the corn, all the genetic information from other ingredients in that food as well, DNA from some bacteria that might be growing on the food. And in the context of that big, messy DNA sample, PCR allows us to zoom in on our region of interest. So for us, that's going to be our GMO transgene, and then make many, many copies of that sequence. So you can think of it as a DNA photocopier. And the way that we achieve that replication is through repeated cycles of a 3-step cycle, and that cycling is controlled by temperature. So the first step of our PCR cycle is denaturation. We want to take our double-stranded DNA and separate it into 2 single strands so that we can read the sequence of bases inside. We do that by heating up our samples almost to boiling, to 94 degrees Celsius. At that temperature, we melt the hydrogen bonds that hold the double helix together, turning that double helix into 2 single strands. The next step is our annealing step. We lower the temperature of the tubes just enough to get our primers to anneal or come in and bind to our sequence of interest. Now, primers are the key ingredient in a PCR experiment. They are short segments of DNA complementary to the beginning and end of our sequence of interest. So they kind of bookend the part of the genome that we're interested in. And then during the third step, the extension step, we raise the temperature of our samples to 72 degrees Celsius, which is the temperature at which our polymerase enzyme is most active. Polymerase is the enzyme that actually builds the new strands of DNA, that actually does the work of replicating our sequence of interest. So at 72 degrees Celsius, our polymerase, which is called Taq polymerase, comes in, it finds the ends of our primers, and then it adds on, building new strands using some loose nucleotides or DNTPs that we've added to the mix. And just like that, you can see we went from 1 copy of our gene of interest to 2. And now when we start the next cycle of PCR, we'll start with 2 copies and we'll end with 4, and so on and so on and so on. And through that process, we exponentially amplify the number of copies of our gene of interest that are present in a sample. And over the course of 30 cycles, which is the length of a typical PCR experiment, we go from 1 copy of our gene of interest to 1 billion copies of our gene of interest. Now, PCR can be a lot, I know. It can be overwhelming if you're just learning it for the first time. And so we've created some free learning resources that students can use to get familiar with that process. If you go to minipcr.com/tutorials, you'll find a great video we have. It's under 10 minutes. It unpacks the science behind PCR, and there's also a great worksheet to go with it. So at this point, our samples are just finishing our DNA extraction, but I want to start setting up my tubes for PCR. So I'll show you what I'm starting with. Before we started today, I pre-mixed the ingredients we need for successful PCR in a set of tubes here. So this tube already contains my primers, my polymerase enzyme, a buffer to keep all the chemicals happy. And I just need to add to this tube my DNA samples. So I'm going to pull them out of the heat block because I think they're done. And as I said earlier, the extraction buffer that we're using is an alkaline buffer. It has really high pH. So before we start PCR, we need to neutralize that, bring the pH down to a more neutral level. And so I'm going to do that quickly now by adding 5 microliters of a neutralization buffer to each tube. And we need to do this because if we don't, the pH will be too high for our PCR reaction to happen the way we want it to. So I'm just going to neutralize this DNA sample. Great. And then now I'm going to take just 2 microliters of my DNA sample from my corn chip, and I'm going to add that to my PCR tube that I've already started to prepare. So you only need a tiny, tiny bit of DNA, and that's because PCR is this exponential amplification process. You only need a tiny bit of your template DNA to start, and then off that exponential amplification occurs, and we go from there. Okay, so I've got my first tube set up for PCR, and I'm going to get the other tubes ready as well. But as I'm doing that, let's talk a little bit more about what we are amplifying today. So what exactly are we looking for when we're looking for the presence of this GMO sequence? So when we create a GMO transgene, we are taking the gene that confers the trait that we like. We're going to move that from one organism to another, but we need to move over a couple other things as well. So in addition, we need to make a promoter a part of this GMO sequence. Now, a promoter is like a landing strip for the enzymes that control transcription. So this is going to tell the new host cell to express the transgene. The transgene's no good if we're not expressing it, right? So we need this promoter here. Now, we want to make sure we use a promoter that's universally recognized by all potential host species as a promoter. And so what we typically use in creating genetically modified organisms is a specific promoter called the CAM-V35S promoter. This is a promoter sequence that was borrowed from cauliflower mosaic virus, and it was borrowed specifically because it's universally recognized. No matter what host plant you put it in, it'll be recognized as a promoter, and it'll cause expression of that transgene, which of course is what we want. We also need to make sure that we have in this GMO sequence a terminator. So a terminator is a signal that's going to tell the cell to stop transcribing the transgene. And again, this is something that needs to be universally recognized no matter what host we try to put it in. So what we typically use when generating GMOs is this NOS terminator. This is a specific terminator sequence borrowed from Agrobacterium tumefaciens, a bacteria, and it was borrowed because it is universally recognized as a terminator sequence. Now, the beauty of the construction of this GMO sequence is that virtually all GMOs have the CAM-V35S promoter and the NOS terminator. So no matter what transgene we're trying to transplant from one organism to another, the content of that gene, the sequence of that transgene will change, but the promoter tends to be the same no matter what GMO we're talking about. So that means we can look for the presence of this promoter, and it'll tell us if a food is genetically modified, is a GMO, regardless of what specific transgene it's carrying. So today we're going to be looking for a 125 base pair segment of that CAM-V35S promoter. We're not specifically testing for the transgene itself because that sequence can differ from plant to plant depending on what trait we're introducing, but this promoter sequence will remain the same. So if we see it, we can be pretty confident that the food that we're testing contains GM ingredients. Now, if we don't see this in our sample, it could mean that our food does not contain GM ingredients, it wasn't genetically modified, or it could mean that something about our experiment isn't working. Maybe we weren't successful in extracting DNA. So to make sure that we can distinguish between these two possibilities in the event that we don't see our transgene, we're going to use something called an internal control. We're going to amplify another gene as a control. Now, specifically, what we're going to be amplifying today is a gene called tubulin. Tubulin is ubiquitous. It should be in any plant, so we should always see it if we're successful in extracting DNA. And we're going to amplify a 150 to 180 base pair segment of that tubulin gene. So it's going to be a different size segment than our GMO transgene, and we're going to use that size difference to distinguish which is which. Now, if we see the tubulin gene in our sample, that means our experiment is working properly. We should always see it if we're successful in extracting DNA from a plant. If we don't see it, that means we were not successful in DNA extraction, our experiment isn't working, and we need to start over. So with that in mind, let's set up our samples. So today, as I said, I'm extracting DNA. I've extracted DNA from my brand X corn chip and my brand Y supposedly non-GMO corn chip. We're going to be looking for both tubulin, our control gene, and our GMO transgene. We definitely should see tubulin for both. We don't know if we're going to see our GMO transgene. That's the fun of this lab. Now, alongside these two samples, I'm actually going to be running PCR on 2 additional controls. Now, when we send you this lab, we'll send you these controls. We're going to send you purified DNA from a non-GMO control and then purified DNA from a GMO control. And these samples will tell you exactly what you should expect to see for a non-GMO food and for a GMO food. So for our non-GMO control, we should always see that tubulin control gene there. We should not see the GMO transgene. And then for our purified DNA from a GMO control, we should see both the tubulin control gene and the GMO transgene. So let's get these samples set up in our PCR machine. So I've got all of my 4 tubes ready, and I'm going to switch to my webcam. Again, we'll open up the mini PCR, and I'm going to add my 4 samples. So starting with my 2 controls, then I'm also going to add my 2 corn chip samples as well.
All right, so our samples are ready for PCR. We just need to now set our PCR program. So I'm going to click back over to the app and set up a new PCR protocol. We're going to call it GMO Lab. And now I'm going to enter my PCR parameters. And this is just a matter of copying over the parameters that we publish in our lab manual. So we've already done the work of optimizing this lab so that you get the most robust results possible in the shortest amount of time. So I'm just really copying and pasting here. And this program should take just about an hour to run. All right. So as PCR is beginning, let's talk a little bit about the next step of our experiment, which is gel electrophoresis. This is how we're going to be visualizing our results today. If you're not already familiar with electrophoresis, it's a way that we can separate charged molecules based on their sizes. And we do that with something called an Agarose gel. This is a dummy silicone version of an Agarose gel. We can use it for practice gel loading, but it looks and feels very similar to the real thing. You'll see that there are there's this row of like rectangles along the top. These are the wells that we deposit our DNA samples into. And this gel looks perfectly flat and smooth, just like the real thing, just like a real Agarose gel would. But if you were to use a microscope to look at the microstructure of an Agarose gel, what you would see looks something like this. It is a spongy and porous, it has a spongy and porous interior. And when we run an electrical field across our gel, it pulls the negatively charged DNA molecules down through the body of the gel. But larger DNA fragments get hung up on these pores and they don't travel as fast as smaller fragments. Now, to illustrate this, imagine that what you see on this slide is our gel. Our wells are along the left-hand side, and those negatively charged molecules are our DNA fragments. The positive pole of our electrical field is on the right of this, and when we turn our unit on, you can see that larger DNA fragments they don't travel as far as small fragments. They get hung up on the pores of the gel, and in the same amount of time they will travel a shorter distance than smaller fragments will. So that's really the principle of how we're able to achieve this separation based on size. So when we are finished running our gel, what we'll see looks something like this. Each of the lines here is called a band. Each band consists of a group of DNA molecules of the same size. When we run a gel, we always run a ladder. So you'll see the ladder in the far left lane of this gel. That ladder is like a sample of DNA fragments of mixed sizes but known sizes. And so when we run that out, we'll see the smallest fragments in that ladder are the furthest band, and then the largest fragments are the bands that remain pretty close to the well. Our samples that we're running out for our experiment are in the lanes next to the ladder. And so when we do an electrophoresis experiment, what we're doing is we're comparing the bands that we see in the lanes with our samples to the ladder to get an estimation of the sizes of DNA fragments found in our samples. So that's how we're going to be reading our results today. Now, we're about to move into loading our gel and starting our electrophoresis run. I had pre-PCR'd some additional samples today that we can use for that step because, as I said, our PCR program will actually take an hour to finish. But before I abandon those samples, that I just started running PCR on. I just want to click back over and check in on them and see how that PCR run is progressing. This is what our mini PCR app looks like when we're running a PCR. It's meant to be really transparent and illustrate what is happening each step of the process. So in the graph you see at the top right of this in the app, you can see temperature is being charted over time. So we're tracking those temperature fluctuations that guide the replication process. In the graph on the lower right-hand side, you can see the number of DNA copies that we've theoretically made for every copy of our sequence that we started with. And then you can see at the center bottom, our animations are showing what is happening to the DNA in the tubes at each step of our protocol. So right now, that animation is telling us we're in the extension phase. The polymerase enzyme is replicating our target sequence using loose nucleotides. And when we switch into a new phase of the cycle, that animation will change as well. So this is meant to be really transparent and illustrate to students exactly what is happening during a PCR run. So we're going to actually leave those samples behind now and continue on with our experiment using the samples I cycled earlier. I'm about to load them into my gel, but before I do, I'll just introduce you to the tool I'm going to be using for electrophoresis today. It is the blue gel from Mini PCR Bio. The thing to know about blue gel is that it combines electrophoresis and visualization. So older generation electrophoresis systems, you would run your DNA out, you do your electrophoresis, and then you might do like an overnight stain to visualize your bands. You'd add a DNA binding agent that would kind of permeate through your gel, bind to your DNA, and then the next day you'd be able to visualize. With blue gel, you can actually mix a fluorescent DNA binding stain into your gel, and it will bind to the DNA as it's running out, so you can see your bands running out in real time. It's nice. It's a system that uses a safer dye than ethidium bromide, for instance, which you might be familiar with. It's a really common DNA stain, but it's more toxic than the fluorescent dye we're going to be using today. I am going to skip a step in demonstrating this lab to you. Usually, of course, you need to cast a gel. You need to make your Agarose gel before you can load it. I did that before we started today just to save time. If you want to know how I did that, we have a video that on the Mini PCR Bio YouTube channel. I've linked to it in the description for this video. In the description below our session today, you can check that out if you're interested in learning more about how to cast gels. There are 3 different methods that video unpacks. The one I use today is using gel green Agarose tabs, which contain buffer, they contain Agarose, they contain DNA stain. You just mix them with water, melt it down, and cast your gel. So with that, let's move on and load our gel. I'm going to switch over to my webcam and we'll get started. So this is my blue gel here on my bench top. I'm just going to take off the lid, and I've already put this, you'll see inside there's a gel, and I've covered it with TBE buffer, just enough to cover the gel. And then I'm going to begin loading this gel on the far left lane, starting with my DNA ladder. Now we send a DNA ladder to you with this lab. It is pre-mixed with a blue loading dye, so that's what you'll see in the tip of my pipette. That blue loading dye, and now you'll see it's been deposited into that first lane on the left. So I'm switching out my pipette tip, and then I'll be loading my samples in the lanes to the right of the ladder. I'll start with my control. Got my non-GMO control first. And you can see where I've added that. Again, because we have a green loading dye that's pre-mixed actually with our PCR components. With the enzyme and the nucleotides that we send you to run PCR, we've pre-mixed a green loading dye. And so you can really take your sample straight from PCR directly into gel electrophoresis. You don't need to add any loading dye or anything yourself. Now I've got my first corn chip sample we're loading now. And then I've just got one to go for my brand Y corn chip. Is going to be my last lane here. There we go. All right. So let's put a lid on the system, and then we'll start the run, and we'll be able to check in on our results in just a few minutes.
All right, so as our gel starts to run, I just want to mention for electrophoresis learners, We have a video and a worksheet again at minipcr.com/tutorials. These are free teaching resources. Do check them out. So let's talk a little bit about what we can expect to see once we can check in on our results in a few minutes. As a reminder, we are amplifying 2 genes in our experiment today. The first is tubulin. Tubulin is our control sequence. It is an endogenous plant gene. It's in all plants, so it should be in all plant-based foods. We're also trying to amplify our GMO transgene, specifically a 125 base pair segment of the CAM-V35S promoter, which is used in virtually all genetically modified foods. So if we see this GMO transgene, that tells us a food has been genetically modified. We're looking for those 2 sequences in 4 samples. So our 2 control samples, our non-GMO control and our GMO control, and then today we're also testing 2 brands of corn chip, our brand X and our brand Y non-GMO corn chip. So when we run out our gel, we at least know what we should expect to see for those control samples. So we should see in both the non-GMO control and the GMO control, we should see that tubulin sequence, that 150 to 150 base pair band in our 2 lanes. For the GMO control only, we should also see that GMO transgene, that 125 base pair band. We also are going to see, we can expect to see some diffuse bands in a really small range below the 100 base pair mark. This is something you see in any PCR experiment. It's called primer dimer. It happens when our PCR primers adhere to one another. We can ignore this. Now, what do we expect to see for our brand X and our brand Y corn chips? We don't know. And that's part of the fun of this lab. We genuinely don't know what we're going to see. It is not a cookbook lab. But we can make some educated guesses about what results we can expect based on what we know about the prevalence of GMO foods. So this is a table that shows the major crops that have been approved for sale by the FDA in the United States. And the first thing you should notice about this table is that it's pretty small. There actually aren't that many crops where GMO transgenes have been introduced. It's interesting to look at why transgenes are being introduced into these crops. We see in the center column a lot of insect resistance, a lot of virus resistance. We also see something called tolerance of glyphosate herbicides showing up a lot. What is glyphosate? It's better known by its trade name Roundup. Roundup is like the major #1 weed killer in the United States, or #1 herbicide in the United States. And the reason why a crop that's tolerant to glyphosate is appealing is because if a farmer is growing a glyphosate-tolerant corn crop, say, they can then spray that crop down indiscriminately with Roundup. Roundup will kill all the plants that don't bear this glyphosate tolerance gene. So all the weeds, anything the farmer did not want growing in that crop will be killed when they spray Roundup. But their crop, the corn, will be spared because it has this genetic modification in its genome. Now, it's also interesting to look at how deeply GMOs have penetrated individual crops. And while there aren't that many crops that bear genetic modifications, the crops that have been genetically modified have been modified to a really high degree. So if we look at this column of the table, we'll see that 95% of canola plants growing in the United States, 92% of corn plants, 94% of cotton plants have been genetically modified. So what does that mean for our experiment today? Well, if 92% of corn plants have been modified and we're testing corn chips, I'm feeling pretty confident that we're going to see that GMO transgene in at least one of our samples. Let's see if that's the case. So we should be able to visualize our results a little bit now. I'm going to flip back to my webcam. So I'm going to take this imaging hood. This comes with the blue gel. This just blocks out ambient light. I'm going to fit it down over the lid of my blue gel. And then I can line my webcam up with the aperture. And if I turn on the transilluminator, great, we see bands. We see bands in all of the lanes where we added samples. So that's great. So I'm going to switch back to my slides now because I ran this experiment earlier, got the same result, and I took a picture that'll be a little easier for us to talk through. All right, here's a picture of our gel. Let's walk through it lane by lane. So you can see our ladder. I've gone ahead and marked the 100 base pair and 200 base pair bands. Everything we're amplifying should fall within this range. Our controls look as we expected them to. So for both controls, we see that tubulin gene. We also see our GMO transgene only for the GMO control sample. Perfect. And we do also see a little bit of that primer dimer along the bottom as well. Now for our brand X corn chip, which remember makes no claims about whether or not it contains GM ingredients, here's what we see. Sure enough, we see tubulin, our control gene, and then we also see that GMO transgene. Not a surprise, perhaps, given that some 92% of corn plants on the market have been genetically modified. Now, what about our brand Y corn chip, which bears this non-GMO project verified label on its packaging? Well, hey, we see our GMO transgene in our brand Y non-GMO corn chip as well. So the GMO sequence is present in a non-GM labeled food. How can that be? Well, let's think about the environment in which this corn chip was manufactured. It was produced in a big factory that produces lots of other foods as well, some of which are GMOs, some of which are not. And so it's quite possible that there are traces of GM ingredients on the same machinery that were used to create these chips. It's also possible that maybe some GM ears of corn fell into the non-GMO corn bin. Cross-contamination could come from many sources. Now, I think that's a testament to how difficult, the context of our food infrastructure, our food production infrastructure, which is huge, which is really complex. It can be really hard to keep streams from crossing, even when you're trying really hard to prevent contamination from happening. And now it's also worth mentioning that electrophoresis is just showing us presence or absence. It's not telling us how much. So we don't know if 95% of this non-GMO corn chip contains genetically modified ingredients, or if maybe just 1% of the corn in these corn chips has been genetically modified. This test is not going to tell us that. And that's, again, a product of the power of PCR. All you need is one copy of that GMO transgene in the sample that you're testing. If it's there, then your PCR is going to amplify and make 1 billion copies, and we're going to see it on our gel. Now, I just tested a couple foods today, but the fun of this lab is that you can test as many foods as you want. And so I'm going to show you now a gel that was sent to us by a teacher who did this lab with their students. And they tested completely different foods than I tested, so let's see what they saw. So here are their controls. They look as expected. They also tested taco shells. They see that GMO transgene in taco shells and in Cheetos and in Doritos. They also tested papaya, which might seem really random next to Cheetos, Doritos, and taco shells, but the papaya is actually, some 80% of papayas on the market in the US have been genetically modified. And that's because most papayas in the United States are grown in Hawaii, and in the 80s and 90s, Hawaii was threatened with the papaya ring spot virus, which was just decimating crops. The papaya crops in Hawaii were rescued thanks to the introduction of a ring spot resistance transgene that was engineered into the papaya crop, and so now most papayas on the market bear that transgene. Now, they didn't see the transgene in their experiment, but they also don't see the control tubulin gene either, which doesn't mean that they were using a non-GMO papaya. It just means that their DNA extraction was unsuccessful, which, as I said earlier, can happen relatively easily for fresh foods. It's a little more challenging to get a good DNA extraction from a fresh food. So I hope my results, looking at my results alongside this classroom's results, should drive home the point that this lab is really flexible, and that's what makes it such a fun lab. You can really take this in many different directions based on how you choose your foods. So a few ideas that we've seen in the years that this lab has been out, you can just do an intense study of one plant. So say you want to go to all the grocery stores in your area and buy up all the papayas you can find and see just how many of them bear that signature of genetic modification. You can select plants in your own garden at home or at school. And this is a nice lab to do if you want to test that claim that I made earlier, that that GMOs are actually pretty restricted. There are only a few crops that actually have been genetically modified. You can put that to the test by testing the plants in your own backyard to see for yourself if GMO signatures are there or not. The same for your produce aisle. Test all the fruits and vegetables you can find in the produce aisle and see if you see evidence of genetic modification. Or you can also expand on what I started today with our brand Y corn chip expose and use only foods that claim to be GMO free and see just how many of them actually for which that's borne out through your experiment. Now, the flexibility of this lab is also what makes it a great choice for independent research projects. So a student can take a semester and use these reagents to test any set of foods that they want to explore. So in summary, hope you enjoyed the lab we did today. Again, this is a wonderful lab that connects to a real-world topic students hear about all the time. In that context, it teaches a complete molecular biology workflow from DNA extraction through PCR and into gel electrophoresis. And it is also not a cookbook lab. It is open-ended, it's project-based, and you genuinely don't know what results you expect to see, which makes it a really compelling lab to walk through. Now, I want to close by answering some questions. In registering for this, you guys asked some really great questions, and I'm going to address those here. So one question we got was about the safety of GMOs. Is it safe to introduce viral DNA or RNA segments into our bodies? Could this lead to production of an actual virus? And I get why that question might be on your mind when you think about what our GMO sequence looks like. Remember, we're testing for the presence of a promoter that is borrowed from the cauliflower mosaic virus. And the short answer to this is yes, it's safe because this promoter sequence that we're looking at is a regulatory sequence. It is not coding for a protein, and the proteins in viruses are the parts that can go on to cause us harm in our bodies. So this is a promoter sequence. It's simply signaling the cell to start transcription. We're not actually creating viral proteins from any part of this sequence. Another great question, can you use CRISPR to make a GMO? You might have heard about CRISPR. It is a revolutionary DNA editing technology that is really changing medicine and changing agriculture as well. And indeed, yeah, you can use CRISPR to genetically modify crops. This is kind of the next frontier of genetic modification. All the GMOs that I listed on that table, they were created years ago before CRISPR was around. So they were not created using CRISPR, but increasingly this is going to be used to do things like silence genes that are causing, say, fast ripening. This is a really exciting frontier because it is CRISPR is a really precise tool. So it's even more precise than the GMOs that we've already talked about. It's kind of like the next generation. It's going to allow us even more control over exactly how a food is being genetically modified. So I hope that addressed your questions. Thanks, guys, for joining today. Before I close, I just want to give a shout out to the rest of our webinars in our series. On May 10th, we have one more left in our spring series. We're going to be talking about a new lab that's up and coming where it's actually very similar to the GMO lab we did today. It focuses on detecting antibiotic resistance in the environment, so you test the soil in your own backyard for the presence of genes associated with antibiotic resistance. So do join us in May. This webinar plus all of our past webinars are available to watch on our YouTube channel. So head to the Mini PCR Bio YouTube channel to catch up on the webinars we've run in the past. And so I want to close there. Thank you all so much for attending. I hope you enjoyed our session.